label it D for dura, A for arachnoid. These little things right here are those, they're referred to as arachnoid trabeculae. Those spiderwebby things, that's the subarachnoid space, the SAS, the subarachnoid space. Okay, and then this is the pia. That line that I drew first is the actual pia. Okay, and so the space between all of these little spider webs is where CSF would be flowing through. Okay, and get a very close-up look at it right up here. Okay, so down at the bottom 154, let's take a look at some more of this stuff about CSF. What else is found in the CSF? Well, it's mostly water, but it's also going to have ions. It's going to have a little bit of glucose, less than blood. Some proteins, again, less than blood. These kind of get filtered by those ependymal cells. If we take a close-up look at this part of the brain right here, over here on your right, this is one of the spaces within the brain that are known as ventricles, one of the spaces that are known as ventricles. And the ventricles have these little collections of little tiny microscopic capillaries. Excuse me. These little capillaries, and that's why they're kind of bright red. And if we look at this zoomed in box, we see the capillaries, they weave back and forth. They make these looping back and forth shapes. And so there's blood, of course, flowing through the capillaries. And then there's a space out here that's filled with this fluid, that's kind of in light blue, that is cerebrospinal fluid. You can see production of cerebrospinal fluid. And these little pink cells, so that cerebrospinal fluid in those spaces then kind of flows down just below the cerebellum, and most of it kind of spills out around the cerebellum and flows around the cerebrum too. That's where we were looking at it on this previous slide. Let me go backwards just real quick. This space right up here, right up across the top, is not really inside the brain. It's actually in this additional space kind of to the outside, and it's flowing right along the surface under the arachnoid, but outside the pia. All right, and that fluid, called cerebrospinal fluid, that is made in the choroid plexuses, which are the capillaries lined by ependymal cells on the surface. Those are called choroid plexuses that are found in the ventricles of the brain. Trying to answer most of those questions that you got down there. Well, really top of page 155. Those choroid plexuses found in the ventricles, they're lined by ependymal cells on the surface that make and secrete and circulate the cerebrospinal fluid. Okay? And it's this kind of dark green, greenish, bluish on this diagram. And if you kind of look at that image from a distance, you can see the brain is actually floating in this fluid. And it's really true. The brain is floating in this cerebrospinal fluid, and it's surrounded on all sides by this fluid. Okay. Part of the cranium or part of the brain? Well, it's not part of the brain. It's not part of the bone. It's really separate. It's really kind of in between. All right. Now, watch as this fluid sort of flows. It's flowing around. It's hard to see on this diagram, but if you got a copy of the textbook, take a close look at it. This is one of those images that sort of takes up a whole page in your textbook, and it's really cool. It flows up over the surface of the brain, and at certain spots up near the top of the cerebrum, if we look in here real close, there are these places where the arachnoid I don't know. I don't have a copy of the textbook. Anybody have a copy of the textbook that you can tell us what page this is on? 154? Okay. So at certain places, this arachnoid kind of sticks out and into this blood vessel called the superior sagittal sinus. And this superior sagittal sinus is collecting blood from the rest of the brain. Oh yeah, I'm sorry. Lecture textbook, not lecture notes. Not page 154 in the lecture notes. I think Cheryl was asking what textbook page. Is that right, Cheryl? Yeah. Page 472 in the big hardback textbook, correct? Yeah. And this is how the cerebrospinal fluid actually gets back into the blood. Remember, check out up here. It comes from the blood originally. It's filtered out of the blood by these ependymal cells, and it flows through these spaces down under the cerebellum, out around the cerebrum, and then right out here, that fluid then gets back into circulation. So CSF is not a permanent fluid. It's not a standalone permanent fluid. It's made from the blood, it returns to the blood, and it only really takes a few hours for it to make a kind of a complete slow-mo circulation around the brain and spinal cord. It's crazy to me every time I teach this and I think about it for 5 seconds, I'm like, wow, my own brain right now has this moving fluid going around it. My brain is floating in this thin layer of fluid, and that fluid is moving, and it's collecting back up near the top of my head, the ridge of my head, along that line between my parietal bones that's called my sagittal suture. It's along that sagittal line, that fluid is collecting again. All right. Here we go. I know it's crazy. It's crazy to me. I don't know if that's crazy to y'all too, but like I said at the beginning of class, it's so weird to think that all the stuff that I teach about, it's not like I teach engineering or math or astronomy, something, beautiful but disconnected from myself. It's like everything I teach about is happening in me right now. It's so crazy. So those ventricles, those spaces, there actually are 4 of them. The one that we were looking at before is this space right here. This is just kind of highlighting the ventricle itself. And that's kind of what it looked like from the side. But if we rotate the brain, if we were to kind of rotate your brain like this so that we're looking at it from the front, that same little ventricle you can see it's actually really thin and flat. It sort of just fills, here it is, just kind of fills that space between the two sides of the two cerebral hemispheres. And it's the third ventricle because it actually receives fluid, cerebrospinal fluid, coming from these other 2 that kind of looks like, they always remind me of ram's horns. They always kind of remind me of the NFL LA Ram helmets, football helmets. So the ram's horns that are the first and second ventricles, called the lateral ventricles. And so fluid flows from them through these little openings into the third ventricle, and then from the third ventricle down into the fourth ventricle, which again, it looks like kind of diamond shaped, but if you look at it from the side, it's super skinny. It's just kind of flat. It's more like a kite, like a paper kite. It's flat and kind of diamond shaped. And this is mentioned in your lecture notes. This one right here, it is in bold. So I do want to make a deal out of it. This tube from the third ventricle that flows the CSF down into the fourth ventricle is called the cerebral aqueduct. You can see in the middle of page 155 of your lecture notes, it's also called the mesencephalic aqueduct. And fluid is flowing in this image from your textbook. The cerebrospinal fluid is flowing in the order in which these are listed. So these aren't listed in a random order. it goes from the fourth ventricle out into the subarachnoid space that we saw on the previous image. That's where it's going from there. But if this cerebral aqueduct gets clogged, it's very skinny, it's very, very thin. If it gets clogged, it will prevent fluid from flowing from the third ventricle down to the fourth ventricle, and that can cause all of these ventricles to swell, and it can actually cause the brain to swell too. So this is another cause of that increased intracranial pressure, increased ICP. Okay, very dangerous, very dangerous. I think we more often see this in infants, both prenatally and postnatally. We often will see it on a 16, 18 week, maybe not a 16 week, but a third or fourth, like a third trimester ultrasound, where, yes, cerebral aqueduct is the connection from the third to the fourth. See my arrows there? It's what flows from it. And if that aqueduct is impaired or blocked, then everything from the third up fills and swells. And so what we sometimes see in neonates, or even before they're born, is a swelling of the brain, but because the cranial bones are not closed yet, the whole head kind of swells. The fluid fills up even around the bones too. And this is called hydrocephalus, which is a terrible term. Water on the brain, or water in the head, is what it technically means. But it's not really water. It's a swollen fluid caused by a blocked cerebral aqueduct. In the past, it was pretty scary and dangerous. It still is. But OBGYNs nowadays have a neat little surgery that they can do where they can actually insert a shunt into the cerebral aqueduct to open it up and let the fluid flow. And if they can control it early on, it's usually not a problem for the baby. It's almost unnoticeable. Anyway, so it flows. That fluid flows. We're still kind of back to the same picture, but it flows from those lateral ventricles that are in the left and right hemispheres into the third ventricle, which is right here. That was the first one we talked about. Through that cerebral aqueduct, pictured right here, that I'm kind of pointing at right there, through that cerebral aqueduct to the fourth ventricle, and then it actually flows out through a little foramen just at the back of the spinal cord, right about at your foramen magnum. You see the bones right here. This is your sphenoid bone, occipital bone. Sorry, this is your sphenoid bone. This is your occipital bone, your foramen magnum. And right about at that level, there's a little opening in the back of the brainstem where that CSF flows out, and it flows through the subarachnoid space, around, out those little outcroppings called arachnoid villi. And the fluid flows out of those arachnoid villi and back into this vessel called the superior sagittal sinus, where it's not really blue. It's actually going to be blood that has been collected from the brain itself and will form the jugular veins on the front of your neck and flow right back into the heart. That's where that blood is going to go. That's kind of how it all flows together. So brain vocabulary, at the very bottom of page 155, what cells create the blood-brain barrier? We talked about that a little bit, not so much last week, but the week before, when we talked about the glial cells, the neuroglia. What cells form that blood-brain barrier? Astrocytes, yeah, the star cells, the astrocytes. What types of substances can pass through the blood-brain barrier? You don't want toxins, you don't want infectious things like bacteria and viruses, but what kind of things can pass through the blood-brain barrier? What things can get in and out of the brain, from the brain to the blood? Think about what the brain needs. What would be the brain's needs? What does the brain need? Brain cells need what? Gotta have oxygen, gotta have, what specific sugar, Austin? I'll put that back on you. What specific sugar that we also know as, yeah, good, glucose. Glucose, oxygen for sure. They need that for their cellular metabolism. In exchange, they also make some waste products. So they have, these waste products have to be able to cross the blood-brain barrier the other way and get into the blood to be taken away. What are the waste products? Yeah, CO2. Excellent. What else? What are some of the waste products? Think about waste products that you guys might have heard of that are actually filtered by your kidneys. Filtered out of the blood, they'll be removed from the blood by the kidneys, but all of your cells make these waste products. I know some of you guys have a little clinical experience and you're familiar with some of these chemicals. We won't really emphasize them until later in ANP2, but they do, the point is they do cross that blood-brain barrier. Many of them are named for the fact that they were originally discovered as components of urine. So one of them is a chemical called urea. One of them is called uric acid. These are just byproducts. They're called nitrogenous wastes. Pretty much all of your cells make them. Not nitrogen in and of itself. No, I mean nitrogen by itself is a gas, and it's not something your cells make. But these are waste products that do contain atoms of nitrogen, so they're sometimes called nitrogenous waste materials. They need to get out and get in across the blood-brain barrier. How about this? This is another one that a lot of people don't often think of, but of all the various chemicals that cannot cross the blood-brain barrier, one that can is ethanol. Okay, what is ethanol? What kind of a chemical is ethanol? We typically don't call it ethanol, but we all talk about it from time to time. Some of us use it from time to time. You can put it in your car. It is flammable. Yeah, it's alcohol. It's alcohol. It's grain alcohol. Yes. And you can put it in your car. You can also drink it. So it's a byproduct of fermentation. And so it can cross the blood-brain barrier. And we know that it can cross the blood-brain barrier because if you drink it, it will affect your brain and it will make you feel buzzed, or it could even make you feel drunk. Yep. That's an alcohol test. That's right. Yep. So these are some of the many things that we know can cross the blood-brain barrier. But I wanted you to think both ways about that blood-brain barrier. Not just stuff that comes from the blood into the brain, but stuff that goes from the brain into the blood too. Waste materials that need to get out of the brain that the blood can take away. Yeah. What about gray matter and white matter? Yeah, got to draw the parallels. You draw the parallels, that's how you remember stuff. Look here. This is a frontal section of the brain. So this would be kind of like cutting it so that you remove the frontal lobe. And you can see that the tissue out here at the surface is definitely darker tan. I don't know why they call it gray matter. It has never looked gray to me. It just looks like two different shades of tan to me. But this is called gray matter, and it's usually found right at the surface. It's always found right at the surface. And this gray matter is gray because it's not white. I know that sounds kind of silly. This is white matter. This stuff is white matter. And what makes white matter in the brain white? What makes it so light in color? What makes the white matter white matter? Why is it so light in color? It actually, yeah, there you go. I was just going to make sure. I was going to say it has to do with what we talked about with neurons. What we're seeing in white matter is myelin. What is the only part of a neuron? What's the only part of a neuron that is myelinated? What's the only part of a neuron anywhere that is myelinated? The axon. Yes, axon. So when you see white matter, you're seeing, if you could look at it under the microscope, you're seeing axons by the millions bundled together. Okay, so this passage of white matter that's kind of whitest on my image, it's just solid axons that are sending action potentials between the two sides of the brain. If this brain was still alive and we could see action potentials, you just see them firing back and forth, back and forth. Okay, here's some white matter going down through here too. This is how the brain communicates with the spinal cord and sends impulses down to the spinal cord. Here's some white matter over here too. Here's little bits of white matter in here. But that's what makes gray matter stand out a little bit. Again, it's not gray, but it's darker because it's not myelinated. It's not myelinated. So gray matter is the unmyelinated parts of those neurons. And what part, if we go with what I've got up here, the axons are the parts that are myelinated. What are the parts that are unmyelinated? What are the parts of neurons that are unmyelinated? What parts of neurons are unmyelinated? What are the parts that never get myelinated? Oligodendrocytes do the myelination. They're not a part. Yeah, the soma. The soma, and what else? Anybody going to come to his rescue? Nope, they're leaving you out there. They're leaving you hanging out there, Tanya. Yeah, soma and dendrites. Soma and dendrites are the unmyelinated. So that's what you're seeing when you look at a cross-section of the brain macroscopically. The gray matter is mostly cell bodies and dendrites of millions and millions and millions of axons right at the surface. Sorry, of neurons, millions of neurons. And then their axons are bundled together in this white matter area. Okay? The gray matter at the very surface of the cerebrum is called the cortex, the cerebral cortex. So that is, kind of draw it right here. That's just like this little bit right there. This little bit right here, that span right there. That's called the cerebral cortex. Yep. No problem. No problem. No worries. Okay. In fact, when you look at the white matter in this picture of the brain on the left, what is causing that white matter is myelination. But remember, what myelinates the axons in the central nervous system are the oligodendrocytes. So you are seeing not individual microscopically, but you are seeing the effect of those oligodendrocytes. But what they're wrapping around are the axons. Okay, so you're seeing the high-speed internet connections between the two sides of your brain. Yeah, they increase the action potentials, yes. Mm-hmm. All right, so that's the gray matter and the white matter. Now the white matter, let's see if I've got this over here. Yeah. So I left that photo, that same photo. So we're seeing the same white matter here, but there's this map that's kind of complicated in your textbook, but it does have the three kinds of white matter tracts that are found in the brain. They're listed in bold right over here, but they're also listed 1, 2, 3 in the middle of page 156. In the middle of page 156, if you guys can see that there. Okay, so the shortest ones, I'm going to cover them in a little different order than they are listed in your lecture notes. I'm going to list them, I'm going to talk about them in the same order that they are in this table over here on the right. And the reason I'm going to talk about them in a different order is because I like to talk about them relative to how far they are carrying information. So some of this white matter that you see up here is actually only carrying impulses from one area of the cortex to another area of the cortex nearby. Okay, so these lumps on the surface of the brain, these lumps or these ridges are called, each one is called a gyrus or gyri, plural. Okay, so the association fibers are little clusters of axons that are carrying impulses from one gyrus to an adjacent gyrus, a nearby gyrus. And so that's kind of what the illustrators in your textbook, not lab manual, but in the textbook, are trying to communicate with those little U-shaped blue lines. And I'm trying to do with my white lines up here. I would say not exactly like gap junctions, no, because we've got whole cells that are trying to communicate, and they're not communicating directly across. They're not communicating directly across. They're actually communicating from one lump to another lump. Now, that we don't want to call them lumps, we want to call them gyrus to gyrus. Okay, so going from one gyrus to the next gyrus. But key idea, guys, key idea with those association tracts on the same hemisphere, from a gyrus to another gyrus on the same hemisphere. Those are called association tracts, or association fibers as they're sometimes called, but association tracts is what we prefer to call them. Okay, they're bundles of axons. That was number 2 on your list in lecture notes, okay? So backing up to number 1 are called the commissural tracts. The commissural tracts, they're listed second here. And the reason why I list them second is because they're a little bit more long distance. They go a little bit further. They are actually capable of sending impulses. They will send impulses from a gyrus on one side of the brain all the way across to the other side of the brain. Okay, that red didn't show up very well, did it? I was just trying to highlight them with a different color. So from one side of the brain to the other, and that didn't do any better. But this is how one hemisphere communicates with cells in the other hemisphere. These are referred to as commissural tracts. They connect hemisphere to hemisphere. Okay, so found between the cerebral hemispheres. That didn't show up well either. There we go. Finally, the projection fibers. The projection fibers are fibers that will go from one part of the cortex all the way down to the spinal cord, or they will come from the spinal cord up to the cortex. So projection fibers connect the cerebral cortex to parts of the nervous system outside of the cerebrum, so to speak. Connect the cerebral cortex to other parts of the brain or spinal cord, other parts of the central nervous system that are not part of the cerebrum itself. Okay? So these are 3 different types of white matter, 3 different forms of white matter that communicate differently. Okay? The terminology that we use now, general vocabulary, bottom page 156, when we have a cluster of gray matter, darker material, gray matter, deep within the brain, that's referred to as a nucleus, a gray matter area within the central nervous system. Remember that it's gray matter, so this is usually cell bodies and dendrites. This is an area where cells are doing the receiving of information, communicating, and deciding on whether an output is needed. So there's a nucleus right there. There's a separate nucleus right there. These are called the globus pallidus and the caudate putamen. Globus pallidus and caudate. And there's another nucleus right out here, right? There's a little bit of a nucleus right down here called the substantia nigra, right there and right there, okay? There's another nucleus right there. Each one of these is a little spot of gray matter deep within the larger brain. It's not the stuff at the surface. That's called cortex. So these down inside are called nuclei or nucleus, all right? A ganglion, I don't have a picture of, but it's a cluster of cell bodies in the peripheral nervous system. Inside the nervous system where we have white matter, these are called tracts, okay? These are referred to as tracts. But outside the central nervous system, those bundles of white matter are referred to as nerves, okay? So a nucleus is analogous to a ganglion. The nucleus is in the central nervous system. A ganglion is in the peripheral nervous system, and a tract is analogous to a nerve. A tract is in the CNS, nerve is in the peripheral nervous system. All right? Then I already got that. There we go. Hold on. We'll get back to you. Yeah, we'll finish with this because this is where, yep, that was where I was wanting to try to get to. So let's finish up with this. The areas on the surface of the brain, I mentioned already that the ridges, the little lumps, are called gyri. The little valleys between them are called sulci, or a single sulcus. Okay. Along the, hold on. Sorry, guys. I had to sneeze, and I wanted to be courteous, so I turned off my microphone so I wouldn't sneeze right in your ear bones. The thing that let me go back because I've got the longitudinal fissure there. You can really only see the longitudinal fissure from a frontal section like this. It separates the left and right hemispheres. The longitudinal fissure separates the left and right hemisphere. So that's that right there. I wanted to go back to that to show you because you don't see it on this side view. It's actually right along, it would be right along up there. What we do see is a sulcus that comes right down the middle called the central sulcus. You can see it up here on this brain up here. It's really like the only sulcus that goes all the way from the top center, longitudinal fissure, all the way down to this little side sulcus called the lateral sulcus. It's the only one that goes all the way down or almost all the way down. And the reason the central sulcus is important is that it divides or separates the frontal lobe from the parietal lobe. Okay, separates the frontal lobe from the parietal lobe. So all this whole reddish pinkish area down there is the frontal lobe. The purple area, even the darker purple, is called the parietal lobe. Okay, but the biggest difference is these key areas called the precentral gyrus. Now I've kind of sketched all over them, so I'll try to do it again. The precentral gyrus and the postcentral gyrus. Remember, this sulcus is the central sulcus. The ridge right in front of it is the precentral gyrus. The ridge right behind it is the postcentral gyrus. Okay? The importance of both of those we'll talk about later, but these are like your body movements that you do with your hands, your feet, your tongue, wiggle your ears, move your eyes around. All of those voluntary muscle movements kind of originate in this precentral gyrus. All of the sensations that you feel on the surface of your body, hot, cold, the sense of touch or pain in your skin, all go back to the post-central gyrus. The reason why those are so critical to know those areas is if you have a patient who has a stroke in one of those areas, they will have, yeah, they're both somatic. The precentral gyrus is somatic motor. The postcentral gyrus is somatosensory, is what it's called, somatosensory. Yeah. So if you have a patient with a stroke in one of those areas, they will have similar symptoms. Okay. Now the lobes, the frontal lobe, this just is the same as the bones that we learned about in the skull. Frontal lobe, parietal lobe, occipital lobe, and the temporal lobe. Okay? But a key thing to remember about these is, I don't want to get into it too much right now. I was going to get into it next week, but I do want to mention it very briefly. I don't know if you guys heard that, but I had somebody trying to call in. To try to disconnect my phone from my computer. That's the most annoying thing. Do not call me during class. So is this. I'll kind of finish with this. This is a weird picture, but we'll get back to this next week. But the right side of the brain controls the left side of the body, and the left side of the brain controls the right side of the body. So, let me go back to this one here. If a patient has a stroke that affects the left side of this part of the brain, this is the motor cortex of the precentral gyrus. So they would have symptoms on the right side of their brain. This is the left side of the brain. So they would have trouble moving a certain part of their body on the right side. If the stroke was just right here, not much further away, but if it was just right here, they would have difficulty feeling sensation on a certain part of their body on the right side, because this is the left side of the brain. Okay, so that was kind of that was my big stopping spot. I'm going to back up here and we'll just finish because I wanted to show you guys some sample questions. Yeah. So here's some sample old questions. What structure makes cerebrospinal fluid? Which of those makes cerebrospinal fluid? Talked about it earlier. Where does CSF come from? Yep, it comes from these structures called choroid plexus that are lined on the surface by ependymal cells. So remember, a choroid plexus is actually capillaries. Capillaries surrounded by on the surface by ependymal cells. So it's kind of like these plus those make up this thing called the choroid plexus. And the ependymal cells will take fluid from the capillaries and secrete it as CSF into the, no problem, and secrete it as CSF into the ventricles. Yeah, no apologies needed. This is where we make our mistakes. We want to get our mistakes out here so we don't make the mistakes on the test, right? Where in the CSF are these structures found? Where are choroid plexuses found? They're found in the four what? Ventricles, yeah. They're found in the four ventricles, right? Mm-hmm. I know you guys are kind of on this because I know that you have not. I know that many of you have not watched the videos yet because you've been studying for exam 3. I'm cutting you some slack on that. No judging, but I wanted you guys to have a chance to see how some of the questions might be on some of this. Collections of cell bodies located in the central nervous system are called nuclei or a nucleus. Remember, cell bodies are what kind of matter? What kind of matter would they be? You only have two choices. Only two choices. Gray matter. Yeah, gray matter. Gray matter or gray matter. I don't care between A and E. Doesn't matter. They are gray matter because they're only cell bodies that are non-myelinated. Woo. Okay, I won't judge you. You don't judge me for my spelling. During surgery, the superior portion of the postcentral gyrus of a patient's brain is stimulated. What is the patient most likely to do or feel in response? All you got to, there's a lot in this question. This is really all that matters to this question. Is postcentral sensory or motor? It's sensory. Postcentral is sensory. Okay? Which is the only one of these things that is a sensory thing, not a muscle movement or a motor thing? Which one of those is the only thing that's a sense? The other four are motor. Does everybody see now? Are you starting to see it? Motor, 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 motor. Okay? So again, I know this happens to you guys on tests. I know it does. You get stressed and you look at a big question like this and it looks big, it looks complicated, but it really is, it just boils down to, do you know what that one part of the brain is for? Okay? So this is a kind of a sample question from an older exam. Do I have one more here? Nope. That's it, guys. We're going to cut it off early this week and kind of pretend that we had an exam in class face-to-face, but you get a little bit of an early leave